to you about life on Earth, but also life in the cosmos. We know that all living organisms have a very basic requirement. All life that we know of on Earth requires the existence of water, requires some kind of an energy source. Now, there are various ways in which organisms can obtain their energy. Plants and diatoms and other photosynthetic microorganisms obtain energy from sunlight, and then they break the water molecule apart, and then they are able to combine the hydrogen and oxygen with the other carbon compounds that they have and make all the necessary proteins and enzymes and DNA and RNA and other life-critical biomolecules. They can also get energy from chemical reactions. We have deep within the crust of the Earth organisms that are called chemolithotrophs, lithotroph meaning rock-eating, and in fact they get their energy from actually consuming rocks and producing the necessary energy and extracting carbon from carbonates in the rocks and making the necessary life-critical biomolecules. And they also need a source of biogenic elements, and the major biogenic elements are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And then there are some trace elements, as we see here, and minor biogenic elements. But all of these elements exist on comets and icy moons of the solar system and in giant molecular clouds. These are essentially widely distributed throughout the universe as a result of supernova reactions. The fundamental question of astrobiology is life restricted to the planet Earth or more widely distributed throughout the universe? There have been a number of very exciting discoveries recently. The most important is that water, we knew it was very widely distributed, but we find that water is present on almost all of the planets and moons of our own solar system, and that would lead us to believe that water would be present on planets and moons of other solar systems as well. So the conditions necessary for life essentially exist throughout the entire universe. This, combined with recent discoveries of microfossils in carbonaceous meteorites, alters the perspectives on the origin, distribution, and evolution of life in the cosmos. The current paradigm, I'm sure what you were probably taught in school, is that life originated on Earth. Earth life originated as a result of lightning bolts in ancient atmospheres and the formation by the famous process of amino acids. And if those amino acids collected in Darwin's nice warm little ponds, then you got the formation of all sorts of biomolecules and ultimately a protocell. Well, there are a number of very, very important problems with this. One of the most difficult problems is that if these complex organics, like amino acids, rain down in the ocean, you have dispersion and diffusion. If you drop some salt in a small glass of water, you can make nice tasting salt water. But if you put the same amount of water in the ocean, it disperses and you are in a huge lake, it disperses and you have hardly any change at all. There have been studies based on genetic complexity of the origin of life from the calculations of the origin of the first genome. And the problem with that, this is studies that were done several years ago showing the genomic complexity, and here about 4.5 billion years ago is the origin of the Earth. But these extrapolations suggest that life actually originated perhaps as much as 9.7 billion years ago. That's a very serious problem because that's far earlier than the origin of the planet Earth itself. This sort of thing suggests that perhaps life didn't originate on Earth. The fascinating problem also, the Miller-Urey experiment does not form all 20 of the amino acids that are absolutely essential for proteins and enzymes in every living organism. Only a small group of them can be formed by Miller-Urey process. And when these groups are formed, they are formed in what are called racemic mixtures. They have, amino acids they have chirality. And so there are amino acids that are like a right-handed glove and amino acids that are like a left-handed glove. Not all amino acids are chiral, but of the vast majority of them are. Well, in all living organisms, all of the amino acids that are present are called the L amino acids, or the left-handed amino acids, and all of the sugars are right-handed, are D sugars. When you have equal mixtures, you have the same amount of left and right-handed, and the problem is that in order for life to exist, it's only going to be using one type of sugar and one type of amino acid. Phosphate is also essential for life because it makes the phosphate backbone of the DNA molecule. And the problem is phosphate was virtually absent on early Earth. The phosphate and phosphorylation process is essential for the genetic code for DNA and RNA, and for ATP, which is essential for the metabolic machinery of living organisms. Several years ago, academician Spirin developed the RNA world hypothesis, and that was the idea that life originated during the period in time when the Earth was frozen, and there formed what was called an RNA world. But the problem is, if the ancient biochemistry of life on Earth originated with the RNA world, or the iron and sulfur world, where, how, and when did the genetic and metabolic machinery change so that phosphate became absolutely essential for all living organisms on Earth? Now, one of the other problems with the endogenous origin of life, life on Earth beginning on Earth, this idea was that once you had these biomolecules, so amino acids and so forth, then you formed a little membrane and you got a simple cell. Well, we now know that doesn't exist. No cell on Earth is a simple cell. This is the DNA gyrase enzyme. This is a bacterial motor. This is a transmission electron microscope image of the motor that operates the flagellum of a bacteria. And if you look, it is absolutely amazing. This is a biological entity with statters and rotors and a shaft. This is the characteristics of a biological motor, and it is essentially identical in all respects to the motor, the electrical motor that was first developed in the late 1800s. So for approximately 4 billion years before the invention of the electrical motor, bacteria had invented the electrical motor, and they had designed it almost exactly the same way that modern electrical designers build an electrical motor. The human genome contains only 3.2 billion base pairs. Polychaeostubium, this is an amoeba, contains 670 billion base pairs. People would think of this and say, oh, that's a simple life form, but look at the genome compared to the genome of a human being. So the point is, simple life doesn't exist. And the other interesting point is phyogenic carbon isotopes and the Ischua-apatites showed that these structures, which appear to be sulfur bacteria that were collected in the Quebec, Canada, indicate that life began possibly as far back as 3.8 billion years ago, which means almost as soon as you had liquid water on Earth, there were living organisms. Well, that does away with the idea that there was this long, slow process of chemical evolution before the appearance of life. An alternate hypothesis is that life originated somewhere else and was delivered to Earth by mechanisms such as the infall of comets, meteorites, asteroids, and so forth. This was originated back in 1871. Von Humboldt said, who could say whether comets and meteors which storm everywhere through space may not scatter germs wherever a new world has reached the stage in which it is a suitable place for organic beings. That is the very beginnings of this idea of cometary panspermia. That life, once it originates, it's picked up on bodies such as comets and then transferred to other suitable places in the cosmos. In 1981, Sir Fred Hoyle and Chandra Wickramasinghe developed this much more thoroughly into the what is now known as the cometary panspermia hypothesis. Many people have said to me, well, that doesn't answer the question of how life originates. It just changes the place. But that isn't really correct, because by changing the place, you dramatically alter the time period available, because we now know that not far from our own solar system, there's a star system that's 10 billion years old. So before life appeared on Earth, only 170 light years away, there was a star system that could have had life that was already almost 5 billion years old before the actual formation of the planet Earth. There are a wide array of conditions within the RNA and iron sulfur world scenarios that are solved by the cometary panspermia hypothesis. And we also know that cometary jets allow the a concentration of solutes and the distribution of solutes and the spinning of a comet as it rotates on its axis. When it gets closer to the sun than the planet Mars, Comet Tempel 1 at the orbit of the planet Mars had a temperature of 67 Celsius, and Halley at 1.8 AU had a temperature of 127 Celsius. The fascinating thing is that the DNA molecule denatures at a temperature between 50 and 60 degrees Celsius. That means the twin helix comes apart. And then when the temperature drops below that, each half grabs the necessary components and recompletes the DNA. And so one DNA molecule becomes 2 when you go above and then below the melting point. And you do it again and 2 becomes 4 and 4 becomes 8. And so in a spinning comet, you could not only have the ability to concentrate the necessary biomolecules, but to do what is called polymerase chain reaction in a natural environment. So this provides not only the mechanism for the origin of life, but an interesting mechanism for the distribution of life once it's formed. Well, many years ago, 1986, along with Sir Fred Hoyle, I published this paper, Diatoms on Earth, Comets, Europa, and in Interstellar Space. And what we were able to show is that the spectrum, this is comet Hale-Bopp, and the spectrum of Hale-Bopp very closely matches the spectrum of a mixed culture of diatoms and olivine. And here's the spectrum of the galactic center source compared to the spectrum of diatoms. And you can see that all the way from 2.7 out to 12 microns in this infrared spectrum, we have a very, very nice agreement between the biological silica that is formed in organic organisms such as diatoms and silicoflagellates and so forth, and the spectrum of interstellar dust. Water and ice is present on all the comets, planets, and moons of our solar system, extrasolar, and by that I mean even the Earth's moon and even the planet Mercury and Pluto. Extrasolar planets are common throughout the universe, and during the last couple of decades we've been studying microorganisms called microbial extremophiles, and we found that they live all over the Earth. In fact, there's enormous concentration of microorganisms deep within the Earth's crust, indicating that there quite probably is more biomass in rocks in the upper portions of the Earth's mantle than in all of the trees and grasses and birds and mammals that you see at the surface of the planet Earth. We have also discovered that microorganisms can remain alive while frozen in ice and permafrost for millions of years, and also in salt crystals while desiccated. They can survive exposure to the hard vacuum of space and to the radiation and temperature changes encountered in deep space. And we found fossils of cyanobacteria, diatoms, and other microorganisms in carbonaceous chondrites. Several years ago, Sabina Buisov, a famous Russian scientist who first predicted that there would be microorganisms present in the deep ice above Lake Vostok, came to NASA Marshall and worked with me on the scanning electron microscope, and we found a host of microorganisms deep in the Vostok ice. This is a pair of diatoms that we found from 2,827 meters deep in the Vostok ice core. These diatoms had just reproduced, and diatoms reproduce by several mechanisms, but one of the mechanisms is standard cell division, mitotic cell division. And you can see here that this cell is a little bit smaller than this cell, and that's because each time the diatom divides by mitosis, the two halves split apart at this dividing line. The parent cell, as it splits apart, the DNA of the cell nucleus splits and goes a half into each half, and then each half builds a new inner half. So this one built a new smaller inner half here, and this bottom one, which was the inner half of this cell, builds a new inner half here. So as a result of that, the diatoms get smaller until they reach a certain limiting size, and then they undergo cell division or replication by sexual processes, and so then they start the entire process over again. Cyanobacteria are most interesting microorganisms. They will cause the atmosphere of the Earth to become oxygenating about 2.4 billion years ago by this incredibly rapid growth of cyanobacteria. Here are two filaments of cyanobacteria that I found and isolated from the stomach of the baby mammoth Lyuba. We studied tissues of Lyuba's heart and lungs and brain and stomach milk. And in the stomach milk, I found these cyanobacteria, which was very exciting because there had been two major theories regarding how this wonderful little baby mammoth died. She was found in Siberia in this incredibly well-preserved state, and she was found in a riverbed, and it was thought that she fell into the river and then sank in the mud and ingested mud and essentially suffocated. The presence of these cyanobacteria in her stomach proved that that idea is wrong, because baby mammoths don't drink water, they drink only mother's milk. And these cyanobacteria are from water, which tells precisely that poor little Yuba died by drowning, not by suffocation in the mud. We went to Antarctica on several expeditions. This is the ice sculptures near Lake Poprudnia. Here I am collecting from the ice sculptures at Lake Poprudnia. These are what we call the ice sculptures, and I put sterile tubes up around little structures like this and broke them off and brought them back to NASA Marshall. If you look carefully, you see little tiny black specks all throughout this ice. These are little black rocks, and they're most exciting because if you examine them in place, even in Antarctica where the temperature is very, very low, the sun shines through the ice, and when they get near the surface, sunlight shining on these black rocks causes them to get hot enough to melt the water, the ice immediately around them. And bacteria, cyanobacteria, and diatoms that are trapped in the ice, now then they're in liquid water and with sunlight, and they start growing and carrying out photosynthesis and producing gases, giving off carbon dioxide. So each one of those little black rocks is a miniature planet. It has an ocean, it has a rocky planetary body, although the rocky planetary body may only be that big, but it also has life living all around these. And the fact that that occurs in Antarctica says it could also occur near the surface of Europa or the surface of Pluto. So there are all sorts of ways in which life not only exists in ice in a frozen state, but actually is capable of growing in ice. These are a new bacteria that we just described in January that we named Sanguibacteria, the Geluda statuarii, for ice sculpture, Geluda statuarii. The interesting point is that comets are considered the most probable parent bodies of the Ci and Cm carbonaceous meteorites. We know from the mineralogy of these meteorites that they had minerals that have been altered extensively by aqueous alteration, by processes within water. They contain clays like the montmorillonite and greigite clays, which a lot of people have theorized was the mechanism for the structure of the formation of DNA and RNA molecules on Earth. But these same clays that are considered important for the origin of life on Earth are also present in the comets, and therefore their role, as was studied extensively with regard to how life might have originated on Earth, is also relevant as to how life might have originated on comets. Vega showed that Comet Halley was not cold, like everyone thinks. When it's out in deep space, it's very cold, but when it gets closer to the sun than the Earth, they measure the temperature of 300 to 400 Kelvin. That's 27 to 127 C, and that's a temperature range that life loves. All sorts of organisms can live within this kind of temperature regime. There were 2 bright jets that were observed coming from the nucleus of Comet Halley. They found 98% water and 1.7% methyl alcohol, 0.41% hydrogen sulfide. This is an image of Comet Halley, and IRAS showed that the albedo was here. This is the albedo of the asteroid 388 Hydeia, and also the Tagish Lake carbonaceous meteorite is shown here. So these are all very black, very dark bodies, which is extremely important because they are so black that they absorb sunlight and get hot as they even are as close to the sun as the planet Mars. The other interesting point, when you look at deuterium and hydrogen ratio, this is the deuterium-hydrogen ratio of comets, and the deuterium-hydrogen ratio of the sea ice carbonaceous meteorites shows that comets fit nicely within this range. But if you look at the Earth's oceans, the protosolar nebula, Jupiter and Saturn, and the interstellar medium, they're very far away, so they would not be considered good models for the parent body of a carbonaceous chondrite, whereas comets have all the necessary characteristics. And this is Comet 9P Temple 1, and here you see at the front edge, the temperature got up to above 340 Celsius. And let's see if I can. This shows the flaring. Oops, well, maybe that little movie doesn't want to work. Oops, well, anyhow, not all technological things work perfectly. If you would have seen that movie, you would have seen all of a sudden this comet getting extremely brilliant. And that flaring is known in comets, and what that is caused by is chunks of the crust blowing off when pressures occur on the interior. And as it explodes and blows off a chunk of the black crust, the comet gets very bright, and then dust and frozen water and so forth is ejected into space, and then the solar wind pushes it out, forming the tail of the comet. On May 14, 1864, at 8 o'clock in the morning, this great, beautiful meteorite called Orgueil fell in southern France near Montauban. There were over 20 stones recovered, and these are cyanobacteria. This is an extremely complex cyanobacterial mat that was present in a freshly fractured interior piece of the Orgueil meteorite. This is just like we see cyanobacterial mats on Earth, large filaments encasing trichomes, multiple trichomes within a common sheath, which is in the cyanobacteria known as microcoleus. This is the cyanobacteria known as Liebia. So there are many different genera and species of cyanobacteria that we've been able to identify within the carbonaceous meteorites. Many people have come along, and if you read about this, you'll see lots of things saying, oh, yes, these are there, but they're only contaminants, because life is present all over the Earth, and so these little bacteria just crawled into the meteorites after they landed. Well, there is a serious problem with that idea, and the serious problem is the following. Lots of scientists have studied all of these different amino acids found within the carbonaceous chondrites, and these are the 8 that are present in the carbonaceous chondrites. The problem is, every living organism has to have all 20. Every bacteria has all 20 amino acids. Every virus has all 20 amino acids. And if I take bacteria and I put the bacteria into a meteorite after it lands, I'm also going to put in not just these 8 amino acids, but the other 12 as well. The 12 missing amino acids absolutely conclusively prove that these meteorites were not contaminated after they arrived on Earth. There's another problem. All living things require all 5 nucleobases, adenine, guanine, thymine, cytosine, and uracil. The problem is, and this is a very important problem, is that we get no cytosine in the meteorites and we get no thymine in the meteorites. Well, cytosine turns into uracil with a half-life of 17,000 years. Thymine turns into xanthine with a half-life of 1.3 million years at 0 C and pH of 7. The absence of both thymine and cytosine says that whatever was present biologically in these meteorites was many times the half-life of thymine, so we're talking about several tens of millions of years in order for all of the thymine to have disappeared. So that implies that any biology that we're finding there is tens of millions of years old, and when the meteorites landed on Earth in 1806 and 1864 and 1969 and 2012, they couldn't possibly have been contaminated with modern bacteria. Now, this is a trichome of a cyanobacteria in the Orgay carbonaceous meteorite, and this picture shows that it had a very thick sheath, but on the inside of this sheath we can see individual cells. This is a cell, this is a cell, this is a cell, this is a cell. So the existence of those cells is very important because it clearly shows that this is a biological entity. And in fact, it is possible to identify the genus and species of this cyanobacteria by the length and diameter of the cells within the sheath. But after an organism dies, the nitrogen slowly comes back out because of the process of deamination and goes back into the atmosphere and then forms into the N2 molecules, which have this tight triple bond. And it is only by these complex metabolic processes that by using the nitrogenase enzyme, that you can break this triple bond biologically and make the nitrogen then utilizable by combining it into amines, which ultimately become ammonia and amino acids, and combining it into nitrates. So then that nitrogen becomes available for living organisms. The absence of nitrogen that I find when I look this same way into the fossils in the meteorites clearly says that those fossils are much older than this woolly mammoth hair because of the fact that nitrogen is completely below the level of detection. This spot here is interesting. We looked at mammoth hair yesterday over at the electron microscope at the Laboratory for Radiation Biology, and Misha will tell you that we got many holes when the electron beam struck the hair of the woolly mammoth because it is breaking down and destroying the proteins because of the high temperature. Well, when we look at fossils in the meteorites, we don't see this kind of a phenomenon. So beam degradation is another way of telling whether you're looking at something that is modern or you're looking at something that's ancient. This shows the nitrogen content of filaments within the Orgay meteorite and Murchison meteorite. We had one specimen of Murchison that was contaminated with fungi, and that was the nitrogen found in the fungal contaminants. These are living cyanobacteria. These are data from Egyptian mummies, from pre-dynastic mummies, from Inca that are 2,000 years old, from Peru. And this is from trilobites and 2.7 billion-year-old cyanobacteria, which we know are biological, but have the same nitrogen content as we typically see in the filaments in the Orgay meteorite. This is that little guy I mentioned earlier. Comes up and ends in a beautiful apical cell here, but when we hit it right there with the electron beam, notice we're getting 45%, 46% carbon, 15% oxygen, but zero detectable nitrogen. That is absolute proof, not just that it's embedded in the rock, which we see here, but absolute proof that that recognizable cyanobacterial filament is indigenous and extraterrestrial. Here in the Orgay meteorite, we see a fossil acrotart, which is known as Leosporidia. There are also heterocysts and large filaments like Lingbia, and then we also find in this meteorite filaments that have structures known as Lophotrichus tufts of fimbriae. These are almost never seen in this way. Well, in fact, I can be more precise. Dr. Rotary Ribka, one of the world's leading authorities on cyanobacteria, told that she had never seen fimbriae like this because when the cyanobacteria or the bacterial cell dies, there's this sticky substance on the outside, and these always stick down in the exopolysaccharide, and the only way they can see them is with transmission electron microscope images. Here I'm seeing them with a scanning electron microscope. They're all through here, and the only way that can be explained is that these dried immediately in a very quick way, and that would be logical if they were growing on a comet and then there was a rapid loss of the water into space. Here we have them at 80,000 X, and you can see that this is a 200 nanometer bar. These little fibrils are about 20 to 40 nanometers in diameter. This is the living Calithrix cyanobacteria from Little White River in Oregon, and it has this structure called a heterocyst. That's where the nitrogenase enzyme is located and where nitrogen fixation occurs. This is in the Orgay meteorite, and here we're seeing basal heterocysts here and here. And this nice filament comes up, and both of these filaments taper. And here are filaments that are actually still embedded deep in the rock matrix. So clearly these cyanobacterial filaments are indigenous to the meteorite, and they also represent recognizable cyanobacteria, and these are complex cyanobacteria with very highly differentiated cells like these heterocysts where the nitrogen fixation occurs. So this picture is absolutely undeniably of biological entities in the Orgay carbonaceous meteorite. This is another beautiful filament. This is a very large filament. And again, here, we took a spectrum here and a spectrum back here. In neither case do you see any evidence of nitrogen, but in this sheath right here, the carbon was about 71% and the oxygen 11%. That oxygen-carbon ratio of 0.2 is the same kind of ratio you expect to find in coal. You wouldn't find that in a living cell. Coal is, of course, very ancient material formed by degradation of biological material, and it is called kerogen. Well, the dominant form of organic matter in the carbonaceous meteorites is also kerogen, very much like what we have in terrestrial coal. These are beautiful diatoms from the Polonnaruwa meteorite. And this is tremendously exciting because this meteorite fell in north-central Sri Lanka on top of a mountain. And here we see a structure called Rimoportuli. The cribra are these little rounded structures here, and the areoli. This is a terrestrial diatom that has been has no organic matter. This still has the organic sheath around the silicate shells. This had no organic matter around the silicate shells. It's how you typically encounter fossils on Earth. But you see these tiny little dimples that we see here in these rings, and the structure of the Rimoportula is seen here. So this is exactly the same species as this diatom filament found in the Polonnaruwa meteorite, and in fact, it's the marine diatom Alococera ambigua, but the interesting point is that Alococera ambigua lives in polar oceans. Now, polar oceans are very cold. The top of the mountain near the equator is not appropriate for life forms from a polar ocean. So it is absolutely certain that Alococera ambigua did not grow in a rice paddy field in Aurangawilla, Sri Lanka. In these extremely exciting Polonnaruwa meteorites, we also find big, very strange things like this. And you see, this is a 100 micron bar. This is a very, very big organism with these gigantic long spines coming out from it. And we see here this organism in carbon, and these paddles and spines are very rich in carbon, but the whole organism shows up in carbon. This is one of the spines, this spine here. Looked at at high magnification. It also has an interesting high concentration of aluminum in this same area. Last May, I think it was May 19th, I was here working with Misha and Anton at the new scanning electron microscope that was installed in the Laboratory of Radiation Biology at JINR. This is academician Alexei Rozanov. And this is the new scanning electron microscope. And to our great astonishment during that time period, this diatom was found embedded in the Orgeii Ci1 carbonaceous meteorite. Now, it actually is two diatoms, because you have this cell and then you have this cell. And this is the smaller one, just as I explained before. This is the daughter cell, and this is the parent cell of this diatom. And it's possible to identify it to genus and species with this Pinnularia sigariana that was first discovered in New Zealand by Neil Fogel in 1955. This is a relatively rare diatom. It's only been described from New Zealand. This is an 8.3 million year old Pinnularia from the Beacon Valley of Antarctica. And you can see it still has its sheath, but it gives you an idea how beautiful this photograph is for the diatoms in the Orgeii meteorite compared to this image, which is from an absolutely freshly contained living diatom, and this, which is a fossil that was obtained from the 8.3 million year old glacier. Here we have in the meteorite Orgeii an array of spines and rhomboids and tubes, structures similar to what are known as alveolata in the Orgeii meteorite. Another interesting point is that the recent space studies showed that all sorts of microorganisms, including tardigrade, cyanobacteria, and green algae, cyanobacteria like nostoc and Sphaerociscus and green algae, were put on these plates and flown on the outside of the International Space Station as part of the Biomex lab. And after they were brought back, after some 455 days, they were still capable of growing and they were still alive. So this experiment clearly shows that organisms can live on the outside of the space station exposed to the radiation and space environment. Well, comets may be the site of the RNA world or the iron sulfur world, and may not only have played a major role in the origin of life, but also in spreading it and distributing it throughout the universe. I want to acknowledge the support of Greg German and John Kramasting, also Claude Baron and Martine Rossignol-Strick and Emile Ladje for the Orgeii samples, as well as Bill Birch for the Murchison samples, and Paul Sapira for both Orgeii and Murchison. And I've had the distinct honor and pleasure for the last many years of working on this research with academician Alexei Rozanov, who for many years was director of the Paleontology Institute of the Russian Academy of Sciences in Moscow, and I had many exciting and important discussions with academician Mary Galimov of Vernadsky and Rotary Virgin of Institut Pasteur in Paris. Thank you very much for your attention.